Voice of the Sea, learning from experts across the ocean. Welcome to Voice of the Sea. In this episode, we're investigating the open ocean food web with oceanographer Jeff Drazen. Jeff has studied food webs since the early 1990s. His work has led to new understandings about the connection between organisms in the deep sea and the more commercially important surface species. Jeff conducts his work with a team of scientists and technicians aboard the Kilo Moana research vessel. The research vessel Kilo Moana is a 186-foot Navy ship operated by the University of Hawaii. The Kilo Moana is outfitted with state-of-the-art science equipment, including 8 laboratories. On this research cruise, Jeff and his team are heading 60 nautical miles northwest of Oahu to Station Aloha. Station Aloha is an area of ocean about 100 square miles where many research projects converge to produce a remarkable collection of observations that are helping scientists better understand our ocean and our atmosphere. Jeff, you're the chief scientist of this cruise. What does that actually mean?
It means I'm in charge.
Oh my goodness.
Which is a little, stressful, but I handle the science side of things and the captain handles safety and all of the shipboard side of things.
And so what's your overall goal as we go out on this cruise?
So the scientific goal is to try to understand the food web in the open ocean out here. What was discovered in the last few years is that there's a really big energy demand for all of these animals living in this deep mesopelagic realm, except there's not enough food. So something is missing.
So you mean there's more animals than the amount of food we've measured?
Right. So there's an imbalance. And what we think might be happening is that there is a different food source. There's lots of detritus in the ocean. And some of that gets broken up into really tiny particles. And we call these things suspended particles. Think of when you get in the ocean, you're swimming in a soup. And so there can be bits and pieces of things you can see with your naked eye, but there's also really tiny, itsy bitsy things, little particles. They don't really sink because they're so small. And we are wondering if this is a food source for a lot of these animals in the mesopelagic, a food source for bacteria, and then ultimately maybe even a food source for small fishes and other animals. The surface waters are what everybody is very familiar with. The epipelagic is what we call it, and that's where most tunas are caught and people see flying fish and dolphins and the rest of it. The deeper ocean, the mesopelagic, is about 600 feet down to about 3,000 feet, and there's very little light there, and it turns out that, well, it's very possible that this zone has a very important role to play in the food web in the open ocean. A lot of animals are hiding there during the day in the darkness, avoiding predators like tunas and billfishes and things like that. And so they do provide food for a lot of the animals we like to eat. And it turns out they're pretty important in terms of moving energy and carbon down into the very deep ocean. So a lot of these animals are constantly migrating up and down. And we can get in and find out, maybe even all the way up to small fishes, whether or not they are eating some of that really little suspended particle stuff. So that might help solve part of this imbalance between how many animals there are and how much food we think is being transported into this zone.
So that's the overall big picture for the cruise.
Yeah. Do suspended particles matter? That's the big question. If you like to eat poke, and yes, then big-eyed tuna dives to 600 meters or about 1,800 feet deep, and they eat a lot of these animals that live in this zone. Same thing with swordfish. They dive to 3,000 meters and feed on squid in this zone. While it is sort of out of sight and unfortunately then out of the minds of many people, these deep layers are really important on a global scale, both for our seafood supply and for understanding the carbon and energy budget on the globe. I'm focusing on the little fishes and squids and shrimp and those kinds of animals. So I tend to study the upper trophic levels in the ecosystem. And so there's another group, a couple of people who are working on the zooplankton, and then there's a lot of others that are working on the small particles. And then actually we even have some people here who are going to be working on dissolved material, actually stuff that's totally dissolved in solution, but is also organic and could be a food source, at least for bacteria.
So everything from bacteria all the way up to these fishes and squids that you're interested in.
Yeah, so it's a nice across food web approach. The next 10 days are going to be rather sleepless. The ship is very expensive. We use every hour that we can, so it's round-the-clock operations. Early in the morning, we're going to be sending down the pumps and water samplers to collect the particles and the dissolved materials. And then a little before noon, probably about 9 o'clock, we'll launch one of the net systems to catch either the zooplankton or the fishes and things like that. That'll come back on board around 2. And then we sort all of that, freeze a lot of those samples, and then there's more water sampling, and then that comes back on board, and then before midnight there's another net tow. And we'll just repeat that for 9 days.
So part of your job as chief scientist was actually setting up that schedule for the crews and making sure that all the jobs got taken care of and the time gets utilized efficiently.
Right. We've packed a lot in, and we've got enough time to get everything done. We have one extra day if the weather goes bad.
Exploring Our Fluid Earth is the dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group. The award-winning Fluid Earth and Living Ocean textbooks are now interactive and online. New activities, updated content, and a teacher community. Exploring Our Fluid Earth is now freely available. Find out more at exploringourfluidearth.org. Welcome back to Voice of the Sea. Next, we are isolating tiny, suspended particles with Matt McCarthy.
So I'm Matt McCarthy. I'm a professor from the Ocean Sciences Department at UC Santa Cruz, and here we are on the Kilo Moana, out in the middle of the North Pacific Gyre. And what we're trying to do is isolate molecules from the dissolved organic matter in the deep ocean. And the general background to this is that dissolved organic matter, these are like molecules that are actually dissolved. They're not particles, so you could never see them even with a microscope. But they actually store most of the carbon, so the reduced organic matter that has food and energy for the ocean ecosystems in the ocean, in fact. But they're so small and they're actually so difficult to get out from all the masses of salt that people know very little about them and how they cycle. So that's what we're trying to do, is trying to understand how that part of the carbon cycle works. So you can kind of think of this as like people talk about it like a capacitor for the Earth's carbon cycle, because there's all this reduced organic material that is sort of stored in the subsurface ocean, and it cycles around and around many times without getting eaten by any bacteria. So one of the biggest questions is, why does this happen? What are the structures of those molecules, or what else is wrong with them that kind of keeps all this energy and food down there? So anyway, those are the kind of some of the larger scale questions. And this thing right here is you can tell home built by all the tubes going everywhere, tangential flow ultrafiltration device. And it's kind of like a little bit like reverse osmosis or desalinization plant that takes sea salt out of water for drinking, except the cutoff on these resins is a little bit bigger. So in there it's maybe about 1,000 molecular weights. That would be like 2 sugar molecules hooked together. And what happens is, all those other footage that you've gotten on this trip of people rushing out to the CTD in teams, bringing up cast after cast, taking huge carboys of water, dumping it. So all that, we did about 3,000 liters from this sample is 850 meters. So that's, in many places, would be about the oldest water in the world ocean, because it's right at the place where the oxygen is at a minimum, and the deep circulation, the conveyor belt that you might have heard of, is very slowly coming up. So, but the problem is, as I said, how do you get them out? And this thing is supposed to do. So it can filter out molecules that are just about the size of maybe 3 sugars hooked together, maybe 2 sugars, so actually not that big. But still, almost all the water or the molecules in the ocean are amazingly smaller than that. And so a lot of what we're doing here is we're trying to go from the whole spectrum. So the water goes around and around, get pumped through the ceiling into this tank, and then get circulated around and around through these resins under pressure. That's the tangential flow part. And anything that's smaller than the membrane actually goes sideways and then goes out the bottom and then comes out these tubes. And then we're putting that into these barrels. We're acidifying it, and then we're trying to catch the really small stuff in this experimental resin setup. So this is the acidified small molecular weight molecules, and this is a chemical resin that we are testing to grab them and hold on to them. And so at the end of this long experiment, one of my students will come in and elute this with some solvents, and hopefully there'll be some beautiful colors that'll come off that'll show us that it actually worked.
The University of Hawaii's Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
You're watching Voice of the Sea. Why is understanding the deep sea important? I mean, I live at the surface.
Yeah, you wouldn't want to go down into the deep sea,
I
not
would
unless
not.
you were in a submarine. The deep sea is the single largest environment on the planet. So if you think about, I think a commonly termed or a common known fact is that the oceans cover about 70% of the surface of the globe. And about 60% of the surface of the globe actually is waters greater than 200 or 300 meters deep. So when you consider the total volume, the deep ocean is absolutely enormous. It's 90% of the living space on our planet.
Oh my gosh.
So we need to understand the function and organization of those systems to really understand our life support on Earth.
And so as the planet's changing and with climate change, that's affecting that deep sea area as well.
It is, in interesting ways. There's a number of increasing threats to the deep ocean. One, as you mentioned, is climate change. The changing climate is going to alter primary production in surface waters. And primary production is all those phytoplankton, all those algae growing. Some of that sinks into the deep ocean, and that's a big food supply for these ecosystems. So if that changes, that's going to have effects that are going to propagate all the way down to the abyssal plains. And then there's a number of other impacts as well. Fishing has changed the populations of surface-dwelling fishes, and that can have effects down into the deep ocean as well. The numbers of predators diving deep to feed in this mesopelagic layer. And climate change also seems to be expanding low oxygen zones throughout the ocean. And that's not something we're studying on this cruise, but as these low oxygen zones expand, they may decrease the total volume that mesopelagic, these deep midwater animals can actually live in.
That living space you were talking about is getting smaller.
It could be, yeah.
And of course, we're interested in that, not just for climate change, but because those fish we like to eat are relying on that deep water area.
Swordfish, big-eye tuna, monchong, walu. There's a long, long list of animals, and they all are eating these critters that we're studying. We hypothesize that the suspended particles are going to become more important the deeper you go in the water column. There's lots of suspended particles throughout the water column, but in the surface waters, there's a lot of sinking material, the marine snow, and the quantity of that declines really fast with depth. So the hypothesis is that the deeper you move in the water column, the more important suspended particles are for the food web.
And you're also mentioned that you're going out now in February and you're going to go back in August. So is there something about that sort of winter and summertime you're looking at?
This goes to the dynamics of the sinking stuff. We know that there's a bloom of plankton in the late summer. And so that's what causes a lot of the sinking material, creates a lot of the sinking material, and it then moves down through the water column and is consumed. And so the cruise we're going on now, there's not a lot of this material in the water column, so suspended particles might be more important at this time of year.
After the research cruise, I caught up with Jeff in his office at the University of Hawaii's School of Ocean, Earth, Science, and Technology. We're here with Jeff Drazen. Jeff just got back from a research cruise on the Kilo Moana looking at deep sea food web ecology. So you went to this one location, Station Aloha, and you sampled everything from these microscopic or smaller than microscopic suspended particles to zooplankton to tiny nekton all the way to the big-eyed tuna, and you're linking them all together.
Yep, it's the whole food web perspective.
And so you guys collected about 200 individual specimens in your mock nest net, is that right?
Well, we collected thousands and thousands of specimens in the mock nest net. What we did is we took aside about 250 to 300 of those and froze them specifically for isotope analysis. So we still have all the other specimens, and those are preserved in formaldehyde, and we can always go back and look at those later if we want. But we had to take a targeted approach because we can't look at thousands, and we'll analyze about 300. These fish right here, those are called bristlemouths, and those are one of our main targets. People have looked at what they eat from looking at their stomachs, and oftentimes there's a lot of mush that you can't identify, and we're wondering if it's gelatinous animals. And these would be little things like salps, or I'm trying to think of things people would see in shallow water. They're not jellyfish per se, but little gelatinous kinds of plankton, and those animals can actually filter almost, well, down to bacteria-sized particles out of the water. These little jellies may be eating these suspended particles, and these suspended particles then are making it through the food web into these guys. So these are the bristlemouth fish here, and you can tell, this is a ruler up here, these are only about an inch long. The eye is right there, and you can see right through them. They have a little bit more skin on them, but they've been beat up a bit in the trawl. And these are the animals that we think may be shuttling the energy from the suspended particles into the larger sized animals in the food web. This genus is called Cyclothone, and this genus is the most abundant group of vertebrates on the planet. There are more of these little fish on the planet than any other vertebrate. There are billions and billions of these. They're never super abundant in any one place, but they live everywhere, all across the oceans. So when you add all of that biomass up and all that abundance up, they're really important. These are small fishes called light fishes and lantern fishes, and a lot of those animals are the most common animals in this deep pelagic food web. Lots of them have bioluminescence or photophores, little light organs all over their bodies, and they form sort of the backbone of some of this food web. And these are a lot of the animals that deeper diving tunas and swordfish and the like will eat as well. So that's the link to us. So this great specimen, it looks like a long eel, and it's got a mouth full of teeth. It's not an eel at all, it's a dragonfish, and this is called the Pacific black dragon. It's sort of a quintessential predator down in the deeper depths, and we think it eats a lot of the migrating fishes, the smaller fishes like lanternfishes and some of the shrimps. And it's really interesting. Underneath its chin, this thing that's dangling down here is a lure. And so the tip of it glows, and that looks like a little glowing plankton to some unsuspecting fish. So, many people have seen the movie Finding Nemo. That was an anglerfish. It's doing the same trick as the dragonfish. A lot of people, when they see pictures of these deep sea creatures and these mouths full of fangs, they're very fearsome looking animals, and they imagine them as being meters long, and they aren't. There's not a lot of energy that gets down there. And so what we think is happening is they take on a very small body size because it takes less energy for them to get to maturity and reproduce. So if you live in a food poor place, it doesn't make a lot of sense to have a really enormous body. You know, these little fishes that we've been looking at in the videos, that's the food supply for a lot of the big predators in the ocean. And so they dive down deep to access this stuff. So there's a direct connection to us and our seafood supply. These are the forage base for tunas, for monchong, for swordfish, a lot of species that we like to eat. These animals, a lot of them, because they move up into surface waters and feed, and then they move back down to depth during the day, they're transporting material, they're transporting carbon. So we're interested in the carbon budget, global climate change, we need to know where is the carbon going, and the fauna, all the organisms in these deep layers may help pull this carbon out of the surface ocean down into the deep ocean and eventually down to the deep sea floor where it would be buried.
So they'd actually help to mitigate or reduce the effects of global warming and climate change.
Exactly. So we need to know exactly what their role is in this. And that's one of the main things that we're trying to answer with this project is we need to know, so how much of their nutrition really comes from surface waters? Because while it's a food poor place, we know that there's more biomass in this layers than the food supply from all the sinking particles coming down. So there's something missing. And so what is that? And we're thinking it's these really, really small suspended particles. And we don't know that, but we can also look at how much of their nutrition comes from all the animals that move up to the surface and feed at night, and that transport mechanism.
So those suspended particles are something people didn't think were important in the overall energy budget of the ocean.
That's right. They were completely overlooked. In any scientific study, you have to be able to replicate. You need to be able to look at something, and then you need to be able to see it again and again to have confidence that it's real. So we tend to focus on those species which are common in our net catches. And bristlemouths certainly are, and we know that they're just common in the environment too. But yeah, nets have biases. Things get out of the way. One thing we don't do very a good job at sampling with our mock nest system are squids. Squids have really wonderful burst swimming when they get spooked. And we do catch some, and we catch pelagic octopi as well, but not very commonly. So there's definitely biases. The problem is, how else do you sample the deep pelagic ocean?
Right.
And so we can take remotely operated vehicles, and we can turn on cameras and lights, and we can drive transects across, and we can count things. The problem is when you turn on the lights, everything scatters. And so they work, and they're really useful. They've been great at allowing us to understand the importance of jellies in deep pelagic food webs, because when jellies get pushed into nets, they just get smashed and broken into pieces. But when you drive transects, that's something that doesn't get scared away. And so that's been a wonderful tool in that regard. So every way you sample has its biases.
And then you just kind of piece it all together to get an
overall picture.
Thanks for watching Voice of the Sea.
The Curriculum Research and Development Group in the College of Education at the University of Hawaii at Manoa. CRDG's training roots go back over 40 years. Through professional development programs, curriculum workshops, research on teaching methodology, individualized school and district training, and so much more. The Curriculum Research and Development Group, improving schools, improving education. CRDG.
Turn your love of the ocean into a lifelong career. Join NOAA, the National Oceanic and Atmospheric Administration, as we unlock the secrets in the deep oceans, track rapidly moving storms, model climate trends, protect and preserve our marine resources, and so much more. It's all in a day's work at NOAA. Find a career that makes a world of difference, enriching life through science, service, and stewardship. NOAA.